way.
So Guy, we picked out a pathway to try and avoid the majority of that fishing gear, and still go along the kind of like the eastern portion of this feature. Nice. And Gary, if you want to stop and take a look at something, just
Yeah,
let us
just
know.
call out what you want to.
Oh, that's a
Bridge now.
large boot sponge.
Let's add one more step. Thank
Let's
you.
take a look at that.
Sure. Aaron, let's push in.
Anybody home? Yeah.
Go ahead, George.
That's what Peter and Ren call it.
Yeah,
Boot sponge.
I was just messing with you. It is definitely.
You don't see me. You don't see me.
It's a little shrimp, a lot of hydroid.
Go ahead.
All right, let's come full wide.
Uh,
She's in data.
Duly noted.
Let's see. So keep an eye out. I'm gonna look down and see what refresh my memory of which species are on the list.
At some point when it's convenient, I'd like to zoom in on some of the white spots on the seafloor to see what kind of
Sure.
organisms those are.
Aaron,
Yeah.
let's push in.
Yeah, that nice, it looks like a cloud sponge of some type.
Yeah,
Hold
so
back.
we've got encrusting sponges on the rocks, but also between the
those
rocks,
anemones.
there are little white specks on the seafloor.
What's that dark, Gary, what's that dark kind of
I think
spongy
that's
like?
just a...
Let's push in
Oh,
all the
wait,
way.
I see.
Is that another sponge?
Dark colored sponge.
Do you want to collect it? Okay.
I can't hear him.
Okay.
Moving on.
Yes.
All right, full wide, Aaron.
Oh, is that desmophyllum? No. I don't think so. I don't think so.
Let me push back out a little bit. All right, Aaron.
Oh, look how pretty.
Why don't you just fly straight? Oh, all right, full wide. We'll come down. And
So if we see any like those moon sponges, kind of like what I always
all
call
right, video
potatoes,
push in.
potato sponges, they want those.
So I
Jamie,
guess,
are these what
yeah,
you're
if we
looking
find
at?
one
yeah,
of
so I guess it's mostly just shell debris.
How deep are we?
210.
All right, thank you.
All right, full wide.
Keep our eyes open, yeah.
Is that the one for cancer research?
Cancer sponge.
Yeah, the one that we were looking for on DFO years ago.
So there's sponges that are involved in cancer research.
Yeah, they wanted like 5 kilograms of it.
Mm.
It's a lot of sponge.
It is a lot of sponge. But apparently it grows in bunches.
Grows in bunches, and if you take portions of it, it'll grow, the rest of it will grow back. You don't have to collect the whole thing.
Bridge now. Add a step. Thank you. Might have missed that one.
There's a lot of different species. All right, we are almost back to the red stuff.
You might get to the end of your tether. We're not continuous here.
There's a lot of redfish down here. Ooh.
Aaron, let's push in.
Can we swing left and get a image of that rock? Sorry, swing right. Sorry, I'm a little
Right.
dyslexic. It's down below.
It's coming full wide.
Oh, it's gone.
What are we looking for?
Oh, there he is. Next to
Which
that sea star.
Yeah, rockfish
Lower.
down there.
Ah, the little guy.
Yeah.
All right, Aaron, push in.
Nice. Now, thank you for turning. That's a young. It looks like a young one.
Juvenile?
Someone's a little camera happy.
Aaron, can we go down and get the entire sea star in with the fish? That's a pretty image. Sharp chin. It's got a
Oh. Get out the
got
way.
Just
shut up.
Oh, can you
Aaron.
zoom out?
Yeah.
Oh, man.
Oh
Okay,
my goodness.
That's the camera happy fish right there.
I was really
thought the
hoping for a
radfish
predation
was-
event.
And away he goes.
He was Lingcod was probably hoping for a predation event too. They're ambush feeders.
Yeah. All right, video, let's come full wide.
I'll make a note in the log that a lingcod just sat on a small rockfish.
Let's hope that makes the dive report. Highlight. Wow, there is a lot of them, huh? This is unnerving.
Nice area, nice
It's
rock.
unsettling.
This is the problem with shallower dives for the data logger. How am I supposed to
There's a
write
line.
all of
Oh,
these
there's a
things
line there.
fast enough?
There's some line there.
Yep.
Okay. So what was that, Jamie?
Well, I was just saying that shallower dives are challenging to data log because the fauna is pretty dense.
Alright, that line looks like it goes wrapped around that rock, so we're...
Yeah.
And then, which way are we gonna head from here?
Once we get here, we're gonna go that way.
Okay.
So
And if anybody knows what kind of fishing gear this is associated with, I'd like to know your thoughts.
Ooh.
Hello.
Wow, look at all these beautiful fish.
Can we see if we can darken bubble up just a little bit? Yeah, that's better.
I'm gonna do one more.
Yeah, sometimes if you go too far, it decides to go all the way to black, so
Yeah.
it's one of those fun features.
bridge nav. Add one step. Thank you.
So I think we have Widow. Let's see if I can get this right, Tom. Widow, Yellow Eye, and I don't know the other, several other ones. Brown, maybe? Oh, beautiful, beautiful sponge. Is that encrusting at all? Is it ridged enough? Gary, do you think if we can hold here?
Yep.
And zoom in?
Alright, video, let's push in.
That kind of, no. Looks like it. Have to find somebody though. Oh, it looks like there's lots of crabs living in it.
Mm-hmm.
Might be squat lobsters, but hard to say. Maybe some squat lobsters and some crabs.
Let's see if we can collect one of these. If you go over to the
Sure.
right a little bit,
There's
it's
that
a
little
little
one?
more raised, yes. Yeah, so this area here, that little loop,
Yep.
looks like it'd be a
Bridge nav.
Alright,
Nice.
full line.
Hold position.
Yeah, you're gonna need to step back some.
Oh, pretty. So let's get lasers on, lasers off. Get the lasers right on the sponge itself.
Bridge nav. Can we have a ship move 20 meters bearing?
Aaron, you wanna push in, keep the lasers
105.
in frame?
Thank you.
Someone from Western Germany
is that
is
one of
tuned
those,
in. Welcome.
Round sponges. Over to
The
the
potato?
left. Yeah, the little potato sponge, the orange one, the little pinky orange one. Let me see. I know they want like the moon sponges, but I don't think they want that one. No.
Talking about
So when you're ready,
that one?
yeah. But no, we're not going to collect that
Okay.
one. But I think when we're
gonna
ready,
collect the other one.
yeah, this nice little lip here,
Okay.
kind of in the center. Yeah, that's perfect focus. There you go. Excellent. Thank you, Aaron.
We have some special
All
props
right,
to
let's
the
come
data
full wide.
logger.
Thank you.
They said I would be reduced to
Would
one
you
fish, two
do
fish,
a
red
step back?
fish,
Yeah.
blue fish.
Yep.
I'm close to that level. So Herc, whenever we do sample, we don't need a large chunk, just maybe like 10 centimeters or
less,
so,
yeah.
or less, yeah.
And that loop.
Just
Do
a
you need me to?
small chunk pulled off.
Do you need me to point it out, Paul? Oh, Paul. Greg.
Great.
I don't think so. I think
Okay.
we're good.
I'm ready.
Someone asked how
Quick
wide
draw.
are the lasers, and they are 10 centimeters wide.
You want to, this is the sponge, Greg.
Yes.
I don't know if you
and
see
that
it.
small.
I wasn't paying attention. Hang on.
and that small little lip that's kind of almost in the center of the screen. Below the green lasers. The
Below
ratfish
the-
is just above it.
Yeah.
Oh, so that right there.
Yep.
Yeah.
Okay. Not the rock, Greg, just the sponge on
Rennie, you
it.
just
I was
want
just gonna,
more
I was just gonna
excuses to use the
try
stick
and
of
roll
pointing.
the entire thing over onto the vehicle.
Jamie's
Is that
getting jealous.
All right, let's see if we settle out a little bit. We'll stay in autos.
Fergus is coming back.
Okay.
Should be at least over you, but maybe, hopefully, a little further east.
All right.
Where is this going?
It's a great question, Rennie. Our forward bow boxes are full, so we don't want to put it there. Both of the starboard, more forward bow boxes are full that are small. So we have 4 available bio boxes. I hesitate to put sponges in the starboard ones because of potential floating, but
What about slurp? Snip and slurp.
I
Low
don't,
pressure.
Yeah, I don't, because we don't know who we're sending this to yet.
Right, they may want the morphology more than the
yeah.
spicules.
so I think we'll either target starboard E, which is the forward large compartment, or we'll have to do starboard C, which is the third small compartment. So maybe we'll get a feel for
It's floating.
its texture while we're pulling it off. Yeah.
Cool.
So Tom is saying the little babies in the sponge are baby sharpshin rockfish. You can see little juvenile young rockfish in the sponge. We won't collect the fish. Well, maybe some of the, just the invertebrates. I think the fish will swim away once we grab onto it. There they go.
And
Oh,
you said we
so-
don't want a lot?
No, we don't want a lot.
All right, we will try for not a lot, but when I touched it, it seems like we might end up breaking off a larger piece.
It's more of a, just the volume. We just need that upper lip part.
Ooh.
Oh, now I see it. Yeah, I see the dilemma.
The sponge will dictate how much we collect of it.
Yeah.
Yeah,
How about,
that's usually true.
let me come up and point to you.
Sure.
If a piece were to break off and float, would it actually reach the surface or no? Question from our audience.
Okay.
Well, we just saw a little tiny piece flake off when he barely touched it, and that slowly floated down, so it's not buoyant enough to go up to the surface. But a small amount of current or thrust could make it float away from the bio box, which is what we're concerned about.
Yeah.
So it wouldn't just rise up and keep going, but it could be close to neutrally buoyant, and so a little bit of disturbance could knock it out of the bio box, and then it would slowly float down.
So I was thinking a little bit more about that sea level question. Sea level was never this low, but that doesn't mean these rocks weren't shallower at one time. These rocks have been moving north relative to the North American plate for 30 million years, and at one time they were a lot closer to land and may have been above sea level.
Excellent update.
Oh yeah, that is.
I saw that catch.
Nice
Yeah.
midwater catch, yeah.
It was recognized. It looks a bit like it'll be
Nicely
difficult
done.
to put in a box.
Okay, that looks extremely floaty, so I'm actually tempted to put it in one of the forward bio boxes. I don't see a lot of associates on it, so I think that'll be okay.
Oh, we can take a close zoom on it.
Is there any sponges in the forward boxes?
One of them has a sponge, one doesn't, so we could put it in the non-sponge side.
Is that lambda or omega?
You want
is
to go to
lambda.
hotel?
But
Yeah.
yeah, a zoom before we put it
Lights.
in to look for associates would be helpful.
And, oh right, I never restarted your thing. You don't have it.
You don't have the button I have.
We didn't restart that GUI, so it didn't take the updated any file.
I still have it under Roman
They're under
Hotel.
Roman Hotel, yeah.
How would you describe that sponge shape, the one we took it off of?
I call it cloud. It's like a combination of encrusting and cloud.
Could we get a greater zoom when you're
Yep.
ready?
All right, Aaron, go for it.
Hi, Lindsay. Good.
And
It's a nice area.
if possible, a quick wrist spin to look at the other side.
Sure.
That's like William's grip on the thing.
Hang on.
All right.
Oh, no. Pull wide.
It almost.
It's on. I think it's on the front porch.
self-sampled.
It's against the
No,
bio
it's-
box.
Yeah.
Is it against the bio box?
Yeah, you can see
Oh,
it in bubble
yeah, yeah.
cam.
Okay, well, we'll go for it.
All right. If we can pick it up again, we can put it in the forward bio box lambda.
Okay. This is one of those no pressure moments, right?
Yeah.
Let's see, everybody loves drunken bubble. That looks stable. Let's do this.
Someone
Yeah, I
says
guess
it's
it's
shaped like an
stable.
old-fashioned cloth diaper. Interesting.
Do you want me to check any of the other arm position before you go, or you got it?
No, I'm pretty good on the arm position. The concern is actually more the camera. Keep it
Yeah.
back.
I don't think you're gaining
Better.
too much there.
Better safe than sorry.
Yeah.
Let's do a
Can
lower
you push
grip
the
force.
sample forward with the bio box?
Yeah, I'm worried about it falling. You know, like if we can always get another piece, but
Nice.
We have a question from someone named Gary, and he says, although you're not as deep as at other times, does the lights from the
Oh,
ROV
okay, it just broke,
affect
so
the
we'll cut
fish
another piece.
that normally don't
Science,
get
we're gonna
any
cut
Okay.
another
sunlight?
piece.
Okay, thank you.
And Jamie, but we are going for that forward box.
Yes, that's still fine.
Okay.
One moment, Gary.
So knowing that, let's, is there anything well, you said there's a sponge, so there might be something floating in that
Yeah,
box
there's a
already.
sponge on the omega side.
Let's try and get the lip again. Yeah. Yeah, that section.
To time the motion of the vehicle.
That looks good. Yeah, good. $32 or 032? Or 0? Oh, okay.
So that's
Yeah,
still
it's
good?
Yes.
Super fragile. All right, go to Herc Hydraulics. And let's open up tool tray.
Tool tray. Out.
Yep.
Oh.
And Jamie, which side, just to confirm?
Lambda, please.
Thank you. Okay, keep coming out. Keep coming out.
You can see the sponge, yeah.
Yep.
That looks pretty weighted.
What else is in Lambda?
There's a coral tucked in the back, maybe a black coral.
Yeah, there's a coral.
Oh, I can just see the little piece of it, yeah.
You could see it in bubble cam.
Mm-hmm.
So a big chunk of it.
Oh, nice.
Nav, that's 032.
032. And you got enough of it? You don't need any more of that sponge?
Nope, that's
I
a
think
good
we're
amount.
good, yeah.
Okay.
Okay. Tool tray.
All right, yep.
Awesome.
Boy, that stuff was super
Go ahead,
brittle.
Data Lab.
Sometimes they just want like the spicules, and so then we could, you know, send that through the slurp, right? So it doesn't matter if it's.
The pieces that drifted off when you were collecting the sample, will they continue to grow on their own when they eventually land?
Depends on where it lands, I think.
All right, thanks, Lindsay.
Okay, where to next?
Yeah. Yeah, where to, Jam? Do you want to keep heading west, or do you want to kind of head over southwest and then southeast?
Oh yeah. Let's
Get a little bit closer.
Yeah, right along that ridge.
Okay,
that,
so we'll get up a little bit more
Mhm.
and then over. Okay. Let's see, where are you? I'm just going to do a 40-meter step at
How far? Oh, we want to go be on top of that?
just
Oh,
a little bit
wow.
further.
Okay.
40 meters at 230.
40
Bridge nav.
meters at 230.
One step 40 meters bearing 230.
Look at that.
Thank you. And then once that's complete,
I think it might be more of the same.
we will go
Yeah.
southeast.
That is a tall feature.
Nice. Lots of small fish in there. Is it possible to zoom in a little closer? Ooh, let's take a look at that lighter pink area right in the view of the lasers, a little up from the lasers now.
All right, short video, let's push in.
Where there's the smaller ones, is that what you're looking
Yeah.
at?
Oh, they're just closed.
Yeah.
Okay.
They just closed up. Fish bumped into them.
Just was really kind of keeping an eye out for the little encrusting corals, the cyanthus. Yeah.
Cool fish.
We have someone named Gary wanting to know if the lights from the ROV, if they affect the fish that normally don't get any sunlight at their depths.
Video, let's come full wide. They don't seem to mind the lights.
But they do run into stuff, but it's not to say they don't run into stuff anyway without them.
Some of the fish biologists think that the sound of the ROV is more
Distracting than the lights, yeah.
Yeah, so they look for ways of using quieter vehicles. But a lot of those vehicles you can't stop, zoom in, look around, and collect samples. So you have trade-offs with counting things, quantifying things versus taxonomy and characterizing. It's a pretty sea star.
And again, just call out if you see anything you want to zoom on.
Roger that.
All right, I'm gonna slide south a little bit so you can bring your heading around to what was the step, 240?
230 is the current step.
230.
Yeah, it's way down here. Yeah,
Got it.
it's a lot of mixed in between. But okay, yeah, come back. Try and come back quickly.
Fish is like stuck in there. See that?
Yeah, he's just resting. Did you see the stack of fish?
Yeah. Fish stack.
Layers.
Yeah. Video, push in. Fish layers.
Ooh, look at
Full
those.
wide.
What type of fish are these?
Those are still the lingcod. Hagfish.
Fishing line.
Fishing gear. Yeah, old fishing gear is usually all weighted down by sediment and biology, but Aaron, push in. And it's coming back. A lot of these little fish have like no fear of the bigger fish.
So are we about halfway along this feature?
Yeah, we're still in the red zone here.
Okay.
Ship's coming to a
Pull
stop.
wide.
I was gonna go southeast. Is that okay? You wanna?
Yeah.
Okay.
Yeah.
All right. After the ship move's done.
We have another interesting feature down this line.
Okay. All right,
We
we're
get
gonna
to
go
before-
13.
Before
1.
11.
We're gonna go 140.
All right, so let's bring our heading around to 140.
Bridge nav.
140.
Yep.
Do I want to keep coming
Can we
this
have
way?
a
Yeah, we'll do that
step
for now.
40 meters bearing 140?
Adjust.
Thank you.
You good? Okay. Randy, how far is it between here and 2G?
Let me double check.
Just wondering how long it's going to take us to get there.
It is 825 meters away.
Okay, so we should probably kind of quickly finish this and move
Right.
on down towards 2G.
Yeah, we'll speed up once we get out of this area.
Okay.
What time do we need to leave bottom by?
We are at 200 meters right now, so
It's just a 15 minutes to the top.
yeah.
Cool, okay. We want to be on deck at noon.
Right,
Yeah.
so we want to be handing over at probably 11:50.
Handing over
Yeah, to
to the deck.
for winch.
So we'll
That
come
gives
up at
them
11:30.
Yeah, that gives them 20 minutes for lunch, and then they go down to the deck. We'll do our thing. We'll recover, then we'll go eat lunch. Because it only takes about 20 minutes usually to recover.
If we can time it, yeah.
Okay.
All right, we're going to drop down of this. We'll lose sight of bottom for just a little bit. And you said 130? 140.
140.
So from a flying perspective, I like to fly with auto heading on, just because that way I know it'll keep the heading. The benefit of that is if I just drive straight forward. Did you see the predation event?
No, missed
I
it.
went after some krill. If I drive straight forward, even though there's different water resistance on either side of the vehicle, I'll still pretty much drive straight. And that I only started really doing that when we started putting on the Niskin bottles.
Nice bait ball.
Video krill.
Wow. Tons.
Bridge now. Add one step. Thank you. I'll track a line once we get to the end of this rocky feature.
All right, let's come full wide, Aaron. Oh, you got some of those in focus still.
Okay, yeah, that's weird. Thanks.
Anyway, everything else we fly manual on a regular basis unless it's something specifically for science. It's time to get up.
Yeah.
Somebody's calling.
And I gotta get a little bit farther out in front.
Yeah, just for the sake of the lines.
Like that one?
Yeah.
A lot of fish visible in Argus cam right now.
That's an 8 shot.
Yeah.
Okay, more fishing line.
Bridge nav. We'll just add one more step. Thank you.
And then personally, I like to be slightly heavy, so I'll adjust my Z-bias so it's just kind of pushing me down. Boy, the chimeras are everywhere. And then, and that way, you know, if I'm zooming in on something, I'll like kind of slowly drift down. You can get a really soft landing, and if you're just barely heavy, then even in like soft sediments, you're not going to push in, you're not going to stir up a lot of dirt.
Yeah.
It's also that every pilot has the tendency of when they zoom in to drive backwards and up. And so that way I know that if I'm like hands off, I'm just going to like slowly settle down. Oh, is that a flag rockfish? I don't know if the stripes at the bottom. I only know like the actual name of 1 or 2 rockfish.
Could we zoom? It just went off the bottom of the screen toward the middle. I've been hoping to see one of those orangish sea cucumbers.
Oh,
It's
okay.
on the right side.
Sure. Video, let's push in. Hold that. And okay, keep going in.
Yeah, awesome. Thank you. Nice golden orange color.
Mm-hmm.
And I think the bright yellow is a sponge. Yeah.
All right, thank you. That's awesome.
It's coming full line. The camera pan and tilt are set for deeper, colder water. That's why they're so jerky. Yeah, that rockfish. What type of rockfish is that?
I think it's a flag, but I'm not positive.
Yes.
A zoom would be awesome.
Video pushing.
Good job, Greg. Greg, biologist.
How pretty.
Cares not about the anemones.
Yes, confirmation from Tom. Flag rockfish.
Nice.
Say it again. What type of fish again?
Flag.
Flagfish.
Flag.
Rockfish.
Rockfish.
Flag rockfish.
Full eyed, Aaron. It's bumping past stuff. Just bump in past everything. Yeah. More fishing gear.
Yeah.
Someone from the live feed would like to know, are these rocks ancient, birch, or coastline?
We're waiting for a response from the lounge.
So there's a lot of different aspects to the control box too. So I have a joy gain. So if I go full forward right now, it'll only be 80% because I'm set at 80. Some people fly at 50, some people fly at 100. Sometimes it depends on what you're doing.
So Brandy, I did hear Guy mention earlier that this is not formerly a coastline, and that's all I know.
Okay, so we know it's not a coastline.
Couple of whelks.
We narrowed it down for you. Thank you for your question.
Video pushing.
Yeah.
Oh, sure, that wasn't even me. All right. Let's see if we can find something interesting.
So Jan, we can get there in about an hour going half a knot.
It's kind of full wide.
Or we can go faster, but then we won't be able to, it'll just be a bit quicker. Faster, okay. Sorry, I can't hear you.
Yes, let's go faster.
Sure, 0.7,
Sure.
okay.
Video pushing.
Bridge now. Can we track a line bearing
It's a coiled
126,
hagfish.
speed 0.7 knots?
It's wrapped around in a little swirl.
1 kilometer. Thank you. One, 2, 6.
Okay.
Those are really cool. Cool polyps.
Yeah. All right, full wide. Let's
Yes.
get back out in front.
It's pretty.
New bearing is 126.
126. Copy.
Should be pretty
So I'm gonna come over.
flat terrain. I think even a little downslope, but just very gradually. No, disregard that. Still slightly up.
Rennie, can you zoom out on your screen? I've lost my high pack for here. Okay. Thank you.
Wow, it is super hard to see me.
Yeah.
So I don't think we'll make it to the end there, this one.
Right. I was just seeing how far that yellow and red feature is.
This one?
Yes.
Yeah, that's about 700 meters away. Are you talking to me, Jamie?
No, data lab.
Oh.
I lost my high pack view from the web page, so that's why I asked Rennie to zoom out. Yeah, our next target is right on the boundary between what's proposed to be opened versus what's proposed to be closed or to remain closed.
All right, Aaron, pushing. Lots of krill. Oh.
Someone asked, do you ever see any whales? Well, Jan just did let us know that they are in this ecosystem, but we have yet to
Aaron,
see
pushing
one.
all the way. That is all the way, isn't it?
We don't typically see them from the ROV
Every now
view.
and then you can watch a
Okay.
piece of krill get stuck on the sea star,
So pretty.
and then it wraps them up. It's come full wide. What are you doing? All right, Aaron, push in. Yeah, there are little krill stuck all throughout
Oh,
that.
that's
Wow.
awesome.
So it's actually catching them.
Wow. So we have this Pycnopodia catching krill, actively catching krill. You see the tentacles going up.
How cool.
Yeah, and then they'll just slowly pass it down to the mouth.
I don't think I've ever seen this before.
I have not.
Jamie, have you seen this before?
No, I think it's very cool. Everybody's been wanting to see a predation event. Well,
Yeah.
there you
Got
go.
it, Kelly.
I thought we had a predation event when that lingcod sat on that
Yeah.
rockfish.
How's the ship move doing? It's not doing anything. Oh, okay. He just has the different screen, yeah. is like a feast.
Yeah, go ahead.
Huh. All right, Aaron, let's come wide. Oh, that was a good shot. He just wants a belly scratch. Let's zoom in on the sea cucumber and then we'll head out. Not eating krill. Too much? Yeah. We'd have to slow down the flow on these. All right, let's come full wide and get moving. Yeah, I feel like the krill got worse the longer I sat here. As I say, if we want any krill, we can just take a Niskin.
I'm gonna do a reset on the fly.
Sure.
We're shallow enough that it should do it right.
This is impressive.
This is
density.
impressive. Yes, I have seen krill like this, but not from an ROV.
Ah, yeah.
Massive.
There are a lot.
This is why we are considered a destination feeding ground for many migratory species. Seabirds, turtles. Well, turtles aren't really eating the krill, but eat the jellies that are in the area. Whales, dolphins. They're all feeding on something that is feeding on these krill. Or they're feeding directly on the krill.
Well, jellies would eat krill, right?
I think jellies will eat anything that
they catch.
that they can catch, yeah.
Opportunistic.
Krillers.
That guy's stuck on the lens. You see that one krill was like stuck like right. I wasn't piloting and it was actually Little Herc, but it had a Zeus on it. And it was at the surface and a sea turtle took interest, and just like because of a weird roll of a wave, the sea turtle ended up smearing sea turtle goop on the lens, and it was on launch, so we had the sea turtle goop on the lens for the entire dive.
That's when you need to employ the T-boss. That'd make it worse.
Yeah, it would probably make it worse. Oh, it was Little Herc, no arms.
Oh yeah.
Aaron, have you guys talked at all about